something where negative correlation can be useful?
Yes, sir. We can see that for if I take the example of chili one, if we take the
You don't have to give example of this. You tell me in general. I want to know. Negative correlation कभी helpful हो सकता है better?
yes, sir, it can be helpful. If we see that some of the traits that which increases the yield, but
Sir, just.
लगा दीजिए नेक्स्ट भी जाएंगे ना सर
इसका पावर बीन का लोकल नाम क्या है?
see
बकला.
Fabici.
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 Right.
you are getting the scientific name alright but
Yes,
belongs
exactly.
to family.
Okay, thank you, sir.
थैंक यू. तुम भी आ जाओ, ये तुम्हारा है, तुम साथ में खड़े रहो. चलो उधर आओ.
Good afternoon, sir. Myself Vishnu Raj. Good afternoon, sir. Myself
ये
Vishnu
मेरा
Raj.
देखा हुआ आप पूछिए.
नहीं सर
यस.
Good afternoon, sir. Myself Vishnu Raj. I am from Prayagraj Swartz. I am today, and this is my work that I've done, and my topic is in vitro screening of chili
11.
genotype under osmotic stress. Sir,
What is
First-
osmotic stress?
osmotic stress means, sir, that
When does it happen? Under which abiotic stress?
जब प्लांट को सर उसके अकॉर्डिंग पानी नहीं मिल पाता है सर, एबिलिटी हो
What
जाती
is
है तो
that in English?
दैट इन इंग्लिश इज कॉल्ड
पानी की कमी.
हाँ
Water deficit.
Water deficit.
Water deficit.
Drought.
ड्राउट.
Okay.
Sir, as we know that sir, chili is most important species and vegetable in our country that are widely grown in India, and we are used in many things, in food, in order, in smell, in taste, in sauce, sir, in pizza, chili flake that we use, sir, and chili powder, or generally, sir, generally during jet season, during jet season, chili in chili that badly affect its germination percentage and productivity. Uh, so sir, so these are sir 28 chili genotypes performed under PG6000, sir, in 2 replications, sir, in 5 stages: normal, control, in 5% PG, in 10% PG, in 15% PG, in 20% PG, and that's the graph, sir. In 5% in control way, sir, this normal, and 5%, sir, this normal growth is okay, similar to normal. In 10% reduce little bit, sir, and 15 and 20% is completely stop. Yes, sir.
So what is the conclusion of your study?
Sir, conclusion is that up to 10% PG is good for drought tolerant. After this, no.
So you used varieties which were from IAVIA.
Yes.
Huh?
25 genotypes you have collected from?
Sir, 25 genotypes collected from the IAVIA. Sorry, 28 genotypes.
28.
Varanasi, IAVIA, Varanasi. Among them, to fight against the drought, we choose. We have to conduct trial on the drought. Okay, so in a field condition, there is a lot of field need. So to minimize this work, what we did, we did little in vitro condition in a control environment. We conducted trial. Then we use PG 6000 in the control 5%, 10%, 50%, 20%. Okay, and we take this five parameters: genetic percent,
Germination percent,
total seeding length, root length, shoot length, and seed vigor index. So in our here you can see the lines. These are 0% and 0%, 5% growth is normal, but when it reaches to the 10% of water deficit, 10% of stress level, this shows a drastic 50% of loss. performance loss. So in a conclusion, we identified the five varieties, five genotypes: Japanese lawn, EC519636, CMB15, Kashiapa, Kashianmo, Perennial, and ISC2. So these five,
tolerant.
And in the next season, we will take this variety in a field condition.
Thank you. But do these things with one person. He should be
Yes,
italic.
in
Okay.
italic.
And mention the family name.
Sure.
Okay. Family Solana. Okay. जी मैडम. जी मैडम.
Good afternoon, sir.
Which number?
Which number?
Evaluation of-
This is second number. This is Kriti Gupta.
No.
So,
Evaluation of exotic light. P13. Okay. Please.
so good afternoon, sir. My name is Kriti Gupta. I'm pursuing my MSc in Genetics and Plant Breeding from Swatch Prayagraj. So my topic is about evaluation of farmers try to cultivate here. So what happens ki the rice variety is harvested in November, है ना? So because of that our wheat like for हम लोग ने evaluate किया था and हम लोग 55 genotypes.
From where you have collected this?
From different sources हमारे HOD ने provide करवाया था, है ना? So we have हम लोगों ने evaluation किया था कि on the basis of certain traits like flag leaf, which is the highly photosynthetic leaf in wheat. So हमने उसका width का देखा था and हमने for example हाँ plant height लिया था और coefficient variance and PCV is phenotypic coefficient variance. If the heritability is high, that is related to matlab if GCV is high and heritability is high in something, in some genotypes, then we can easily transfer those traits to make new varieties which are stress tolerance, है ना? So majorly हमने दोनों conditions में लगाए थे, late sown and timely sown. So timely sown में high GCV and with high heritability was there in flag leaf width, and late sown में हमारा high GCV and high heritability was there in plant height and plant like grain yield and also with the SPAD reading which is for chlorophyll content, है ना? So मतलब these characters we can use for making different stress resistant varieties and all. So this is, thank you sir. Yeah.
Okay.
Yeah.
Just a percentage. You have done these things in replicates.
No, augmented.
No. I think suppose you are taking 55 varieties, this days for 50%
Heading,
and
yes.
these things, no?
Yes,
These
yes, yes.
characters you are taking.
Yes.
You are taking, whenever you are doing the experiment, you keep prepare all these things in the replications, no?
Yeah.
So please give it to the standard error. Not giving the standard error, then it is meaningless. Okay.
Okay.
That's everything is fine. All the best. And another thing, always mention belongs to whatever the, not only wheat, any plant specimen you are doing, you have to mention the family names. Okay, because it is rules.
Okay.
Okay. Thank you.
It was my first time, by the
Okay,
way.
all right, no
Okay,
problem,
Thank you.
no
Thank you, sir. Yeah.
भाई এটা খুলিব কি What's your serial number?
Sir, it's I guess 10.
10, no? P10.
Yes, sir. And this one. Sir,
Yes, please.
sir, it's role of morphological trait in
Yield expression. Yes. Proceed,
सर
proceed.
इस सर आपको क्लियरली बता दो हिंदी में बता दो
yes, yes,
ओके
yes.
सो इसमें सर चिल्ली के इल्ड के लिए करा गया था जिसमें 40 जीनोटाइप्स दिए गए थे
40 genotypes.
यस सर 40 जीनोटाइप्स और इसको रैंडमाइज ब्लॉक डिजाइन में 3 replications में लगाया गया
From
और
where you have collected the 40
सर
genotypes?
IIVR
Varanasi.
यस सर
Okay.
सर और इसको रैंडमाइज ब्लॉक डिजाइन में 3 replications में लगाया गया था और उसके इल्ड को एग्रोनॉमिक ट्रेट्स और इल्ड के बेसिस पे रीड किया गया था और जो
Heat map. This
यस सर और इट वाज जेनेटिक एंड फिनोटाइपिक बोथ सो सर जब जो रेड वाले साइंस हैं वो पॉजिटिवली कोरिलेट करते हैं और जो ब्लू वाले साइंस है वो
Negatively
नेगेटिवली
correlate.
सो दिस इज द डाटा और फाइनली इसका कंक्लूजन ये निकलता है कि इफ
These are the various traits.
yes sir these are ill traits
Ill traits.
like fruit per plant fruit per plant or weight of fruits or leaf and stem diameter etc और सर इसका कंक्लूजन ये था कि फ्रूट मतलब कि स्टेम डायमीटर फ्रूट पर प्लांट और प्लांट जितना स्प्रेड होगा तो फिर वो मतलब उसमें लीफ ज़्यादा आएंगे और क्या बोलते हैं उसको फोटोसिंथेसिस ज़्यादा होगी सो दैट उस पर ज़्यादा इल्ड ऑब्वियसली आएगी और
You have taken the photosynthetic parameter here?
यस सर
By SPAD or anything, you have calculated the photosynthetic parameter?
सर नो इट वाज एवरीथिंग वाज मॉर्फोलॉजिकल ओनली ना
Only morphological,
यस
no?
सर ओनली फील्ड
Only field based data. Okay, all right, thank you. But all the fellows I'm telling, mention these family names, okay?
ओके सर
Solanacea, this belongs to which software you are using for this?
सर आर
R software. Okay. All right, thank you.
थैंक यू सर
Your poster number, please.
0P3.
3.
Samira Mansoor.
Please.
Good afternoon, sir. I am Samira Mansoori from Government Holkar Science College, Indore, Madhya Pradesh. I am representing my research on suggesting hydroponics as a viable alternative for growing medicinal and aromatic plants. So, for understanding what is the scope of hydroponics in the field of medicinal plants, we did a literature survey, and then we identified few major research themes, which were plant growth and physiology, targeting the metabolites and the nutrient management. That is a key aspect when we are working with the hydroponics. So hydroponics is basically a soilless cultivation system in which we do not use the soil, we eliminate the soil and replace it with the growing media such as cocopeat, clay balls, perlite, vermiculite. Now all these things are actually the waste products or one of the another industry. So then we selected two model plants, mint and rosemary, and now we are working on, then we designed a system for growing these plants. So we found out that mint grows particularly well in the Dutch bucket and indoor hydroponic designs. And since rosemary is a shrub like plant, so hydroponics are mostly used for growing the herbal plants like spinach, lettuce, and these kind of small size crops, herb crops. But rosemary is a shrub plant. So we needed a system that can be viable for it for at least 12 years, because rosemary is oil that we extract from the rosemary can be has a 12-year cycle. So you can use that plant for 12 years and extract the oil. So we designed a Dutch bucket design for it. There are 3 Dutch buckets which are connected with a reservoir. In the reservoir we add the nutrients. For nutrients we use Hoagland modification.
Hoagland direct, or you have modified something?
So Hoagland modification, that's all. We have not identified, but I'm pleased to announce that we got good result with the Hoagland modification media only. Now we are working on enhancing the metabolite production. So this and we have a grow light connected with it for the rosemary. So as the plant grows up, the grow light also goes up. So the grow light can be shifted. So we don't have to worry that we have to take out the plant and plant it somewhere else. The whole system supports the rosemary. We found out that in the hydroponics, rosemary had more phenolic compounds, and even when we grow mint, the hydroponically grown mint had more flavonoids and phenolic compounds. So now we are targeting on working on the nutrient so that we can enhance this metabolite, and then we can use it to for the industrial purposes.
Okay. Just let me know one thing. You have done these things, or you have gone through only the review paper and prepared?
Sir, I have done these things. So since I've already presented my research on mint and rosemary somewhere else.
Okay. You are MSc or what?
Sir, I'm PhD, research scholar. This whole is my PhD work. So I'm working on 3 plants, mint, rosemary and lemongrass. The lemongrass one is still on hold, and these 2 have gone for the phytochemical analysis.
Phytochemical analysis. What are the phytochemical analysis you are doing here?
So we will be doing HPLC. HPLC and GCMS for the essential oil content. I mean, target the essential oil. Because rosemary and mint both are used for the menthol.
You have extracted the essential oils?
Sir, I have not yet extracted the essential oil. This is the beginning work.
Okay. So first you will go for...
so we reviewed and then we found...
No. I just want to know for essential, for that phytochemical analysis, any component you got, any specific chemicals? Or
So not yet. We did a total phenolic compound.
Total phenolic compound.
That was
qualitative
higher.
Or quantitative?
So we did using spectrophotometer.
That is the
quantitative analysis. So we found out more phenolic compounds. Now that since our college HPLC unit wasn't working, so I couldn't get that data now.
So just one thing, if you are going for any that phytochemical analysis, you can do the you will do this spectrophotometric analysis for your quantitative analysis. But if you will go for the HPLC analysis, if in the HPLC analysis is like that, if number of standards are present there, just like camphorol, acetone or anything, okay, so that you will get a good result, and you will get whatever you will get so many compounds if according to the it is dependent upon the standards present in the HPLC. If there is not much of standards are present in the HPLC, then you will not get a good result. Okay, so it is better you can go for the HPLC. It is my personal opinion. If you go for the GCMS or LCMS, volatile and non-volatile compound, it is better for you.
Sure, sir.
Okay.
Because that is remaining now. So thank you for the feedback.
Sir, myself P. Bhavna. I am a PhD scholar from OUT. I have conducted my research work at
Madam,
CRRA.
P16.
Yes, sir.
In CRRA you are doing work?
I am an OUT PhD scholar and I have conducted my research work at CRRA Cuttack.
Under whom?
Under Shyamaranand Das Mahapatra, sir. and Totanadak as my member. So sir, as we know, the rice leaf folder is a major defoliator in rice, and in order to control this pest, we have been using higher doses of, we are depending on chemical pesticide. This chemical pesticide has led into pest resurgence, increase in pesticide resistance, and various environmental pollution. So we have thought of coming up to some using or exploitation of plant induced defenses, out of which herbivore induced plant volatiles are environmental friendly and sustainable. So based on this, preliminary studies I have done, in which screening of 52 red rice accessions was done.
Red rice you have collected from the gene bank of
CRRI. And based on 52 red rice accession, 3 field screenings and 2 net house screenings were done.
3 field screenings and 3 seasons.
3 seasons, yes.
So consecutively in Rabi or Kharif?
So 2 Kharifs and 1 Rabi.
Okay.
And 2 Kharif for net house screening.
Okay.
And based on that, we have found 2 highly resistant varieties and 2 highly susceptible variety, along with 1 susceptible check, that is TN1, and highly resistant check, that is TKM6. They taking all the 6 varieties, we have taken
one interruption. You have taken susceptible TN1 and resistance
TKM6.
In between that is moderately susceptible, moderately resistant.
No.
Only susceptible.
So we have
and
taken
resistant.
2 checks.
Okay.
And 2 from the field screening which we have done, 2 resistant and 2 susceptible.
Previously these 2 are reported, TN1 though I know, but
TN1 TKM6 is reported, sir.
previously reported by ECRIP.
Yes, sir.
Okay.
Even ERI, sir.
ERI also, okay.
And based on my 52 red rice accession screening against rice leaf folder, I found these 2 varieties, Kalobath and Koka is highly susceptible variety, and Kalibank and Asapal is highly resistant variety. Combinedly taking these 6 varieties, I have done the volatile extraction. Volatile extraction was done in both the healthy conditions, that is uninfested conditions, and after infestating with rice leaf folder for 4 hours, then volatile collections were done. And after the volatile collection, those crude volatiles were injected into GCMS machine.
Just let me know, in the net house condition, how you are infecting this rice leaf folder?
Sir, in net house conditions, in the 45 days of rice plant were covered with mylar cage. And in one pot we have 4 seedlings. And we are releasing 2 second instar larva. Yes, sir. In larva and allowing to grow. And after 15 days, we are taking out the mylar cage, and the damaged area is being taken over the graph sheet. The area or the in mm square, we are observing the damaged area. In field conditions, we are doing as per the ADLS scoring, the reported by CES scoring of ERI. In field condition, we are taking number of damaged leaf by total number of leaves.
Yes. Okay.
Yes, sir. Then the crude volatiles that has been collected from both the infested and uninfested conditions, from both the susceptible and resistant varieties, were injected into GCMS machine. GCMS machine has separated all the individual compounds, and it has shown at the based on boiling point and our molecular weight.
All right.
Yes, sir.
No problem. Then what's the result you'll get?
After that, sir, we have seen that the terpenoids, the aldehyde
coordinate analysis.
This is PCA metabolomics, sir. Based on that, we have observed that the terpenoids, the aldehydes, these are highly induced in resistant varieties, and after infestation in resistant varieties, these aldehydes and especially terpenoids are more induced. And we can clearly observe whereas uninfested susceptibility lies in the right hand side of the PCA, whereas uninfested resistant lies in the left hand side. Based on that, we have done the PLSDS scoring. In VIP scoring, we have observed that in VIP scoring, more than one are the compounds which are causing the main discriminatory compounds, causing differences in whether it is susceptibility or resistant. So based on that, methyl jasmonate, limonene, xylene, trans-betafanine, these are the compounds. These are highly enriched in infested resistant and in uninfested resistant compared to susceptible. So based on that, we have found that 50T total compound from MS. These compounds, for example, sir, as because of space, less space, I have just given, we have done GCAD, gas chromatography, electron-antinography, in which this is insect gives negative deflection, whereas GC gives positive deflection. So when we are running the GC, and if this insect is responding to those compound at particular RT, it will give a negative deflection. So in case of uninfested susceptible variety, we have found that 31 compounds, the antenna is showing response. Similarly, combining all the uninfested resistant, infested resistant, infested susceptible, we have found in total 42 common compounds. So 42 compounds cannot be taken for further behavioral studies. So we have to screen it down. So for that we have done electro-antennogram, in which in a Pasteur pipette the individual pure compound was injected and it was observed. And based on that, higher EAG amplitude was observed for alcohols, next aldehydes, then
terminal heat stress. है ना? So generally in India, farmers what do try to do is they want to sow or cultivate both rice and wheat in a year. है ना? So because of that, what happens ki they harvest the rice in November, and then the sowing of wheat gets delayed, due to which they face terminal heat stress, and which is bad for our yield. है ना? So for that we have done some studies. We have made a study on 55 varieties. 55 genotypes we have taken, and we have done study on that, and we have selected some traits like flag leaf width, which is the highly photosynthetic leaf, which does the high photosynthesis in the wheat, and there are some other characters like plant height, and SPAD reading, which is for chlorophyll content, and we can say grain yield, peduncle length, different traits we have selected. So generally what we have observed is we have sown these varieties in both the conditions, which is late sown and timely sown. So in timely sown, we have observed that flag leaf width is genetically more heritable and have higher GCV, and as well as it has higher PCV. Yes. GCV is genetic coefficient variance. So it has higher GCV, as well as it shows high heritations. SPAD reading has high heritability and high GCV, as well as in plant height, it has higher GCV and heritability. So these characters majorly we can use for selections, simple selections, and breeding programs to for breeding and to release new varieties, good varieties for our farmers, so that they don't face these kind of problems. What's your opinion?
Thank you.
Thank you, sir.
Generally, yeah.
Based on which characters? Only the physical characteristics, no other genetic characteristics.
Yeah, genetic characters we see, but sir, I am talking about the physiology especially. So, if I talk about physiology, then
Physiological character, you think that physiological character is heritable?
yeah, why not? Some are no.
Some are.
Yeah.
But
not some are there, but not every character. As I already told you, like the flag leaf width is important for photosynthesis. So
right.
Yeah.
But flag leaf height or flag leaf width, is it permanent character for all type of soil? Not that.
No. Yeah, exactly.
That is why it is not heritable character.
Okay.
Only you can tell. Let's say that only genetic character may be heritable.
Yes.
Stable character.
Yes.
Then you have to go for genetic.
Yeah, exactly. Yes, you can go for that.
That's right.
Yeah. Some marker assisted selections. Yeah, we have to go for that. So we are trying. I'm very new and initial student. Yeah. No, I'm master's student.
Masters.
Yeah.
So they masters dot.
Sorry?
That institute masters in the already, and they have
I am first year student.
in that institute.
Yes. Yeah, I'm first year student.
That is not only for research institute.
No.
Oh.
Thank you, sir. Oh, sorry.
Sir, my presentation is role of morphological trait and yield expression in chili. Yes, sir. So for this,
Just objective of this work.
so genetics and plant breeding.
Objective. What
Objective. So, sir, my objective was to correlation coefficient with मतलब so yield analysis and yield attributing traits. मतलब sir, if you work in yield with agronomical trait, so like can I using Hindi also? So, sir, if we have good fruits, good fruits, then yield will be increased.
Right.
If we have a strong stem, when the chili plants stem will be strong and broad. So if it will,
bigger vegetative.
yes sir, so it will be it obviously increase the yield. So that's all this presentation is. So for this we have selected 40 genotypes from IIVR and randomized block design.
You select that genotype on the basis of
Sir,
their physical characters.
Yes, sir. We have taken these varieties from IIVR for only morphological study of this, like for yield expression.
On the basis of yield expression.
Yes, sir. Yield
You
expression.
selected that
40 genotypes. Yes, sir. Or we have in sir, मतलब कि 40 genotypes को RBD में फील्ड में लगाया एंड देन 3 replications हर प्लांट के और उसके बाद सर
procedure, that means manual.
is
All the same.
not we have all the same.
Is there any difference in field condition?
No, sir.
No.
Like we have managed कि क्रॉस ब्रीडिंग ना हो, सेल्फिंग हो, लाइक उसके लिए पूरा प्रोसेस ताकि उसको कवर करके रखें.
So that field condition, I mean manual.
Yes, sir. Field has
condition
been
is the
prepared. Yes, sir. Field
There is no input or something condition, not that.
No, sir. Field condition was same.
Was same.
Yes, sir.
In same field condition, input the
40 genotype to see the yield of different genotype, which are like देखना ये था कि कौन सा जीनोटाइप ज़्यादा अच्छा वर्क कर रहा है, कौन सा ज़्यादा अच्छा yield दे रहा है, because it's all about yield for the farmers. Because
after screening that, you will just select.
Select the superior and then we'll grow it. And we'll work on later on. We'll work on to release some variety if we get best from this.
On the basis of
Sir, no, it's not phenotypic. No, sir. It's not about it. Like now I am in first year, sir. So we are working on this, and after that we'll work on genotypic things also.
Yes, you have to study
Yes.
phenotypic characteristics.
This is only.
Phenotypic characteristics is the permanent character.
Yes, sir. This is only just for the understanding of farmers and
changeable according to the environment.
according to soil also.
Soil condition, environment, other parameters.
Yes, sir.
That means you
And
have to genotype.
yes, sir. Thank you, sir.
So next,
Sorry.
Yes.
First of all, just tell me that what is your objective of
So the objective of my research was to actually find out which hydroponic systems work well for the medicinal and aromatic plants. So we are basically targeting mint and rosemary. So we have found out that hydroponics is basically used for leafy vegetables and plants like that. But for medicinal plant, it is slightly a newer concept. So first we find out that each kind of hydroponic system is designed for a different kind of plant. For example, we cannot actually grow tomato in its pipes. Correct? We will need a different kind of setup for that. So same thing we applied for the medicinal plants. When we found out, so firstly we did grow mint in the vertical towers, but the mint has bigger roots, so the roots got entangled and it was very difficult to extract the plant. So we do not want that kind of difficulty in our system. Then we shifted to the indoor hydroponics. Okay, and we have designed this. This is a Dutch bucket system. There are 3 buckets which are connected with the reservoir. The reservoir uptake the nutrients through drips. So each plant is given a separate nutrient through a drip system. Same design we use for rosemary as well. For rosemary, the light is adjustable because rosemary is a shrub. So as the plant grows, the light can be moved up. So that won't be a problem. So basically my work targets designing the system for the medicinal plants hydroponics. Now I'm targeting enhancing the metabolite production. While when we grew mint, we found out that phenolic.
What is the mechanism of your production varies from plant to plant? Some plant you call indoor hydroponics, some plant.
So mint is a herb plant. So mint is going good in indoor hydroponics. And the rosemary shrub plant, we designed one system. We tested 2 plants on it, mint and rosemary. Both of them work well. Okay, we designed one system, we planted 2 plants. But initially, since it was new, we also tried growing mint in the Newton film technique. We also tried the vertical towers. Mint also grows downwards also. Roots के side से भी रहने वाले हैं, suckers रहते हैं. So those are not easily extractable from here. We can extract them easily when we have a fixed kind of volume. So that's why we designed a different system. So initially, since there was a gap in designing the systems, so we have designed the systems. Now we are growing plant in these systems. Now we are targeting for enhancing the metabolite production so that we can target the industrial market for these plants.
What does it explain?
So this is a keyword co-occurrence network. So there is a software called VOS Viewer. What we did was we extracted all the research papers from Scopus. Then I did the keyword extraction. These were my major keywords. Okay. Then we did a bibliometric analysis using a VOS Viewer. So we got a cluster map. Now through this map we can say hydroponics was linked to multiple kinds of studies. So after analyzing these and studying all the papers that it was connected to, we found out our major research themes. After that, we have worked on the.
What does it mean that different color?
So these are different routes. So if my main keyword is hydroponics. Now if there is a paper published on hydroponics, it is published with another keyword which is plant root. Then it is interconnected to another one which is soil, and then to cadmium. So you can actually connect the keywords and make a map and understand which kind of study. Like if you are studying hydroponics, so hydroponics for medicinal plant is connected here. For medicinal plant, what kind of things are working on? So for medicinal plant, you can see essential oil and chemical compounds that we are targeting. So this map actually gives you a kind of direction of what research already has been going on. So you actually don't have to read all the papers. You can just filter out certain papers through this. So now I'm working on enhancing the metabolite production for all these plants. So through my research, I suggest hydroponics as a viable alternative where water availability is low and soil has been degraded, because our systems use 90% lesser water than soil. Although the plant grows in the water, you can say basically, but the nutrient is recycled. All these systems are recycled. तो plant को जितना uptake करना होता है, plant उतना uptake करता है, remaining gets into the reservoir. These reservoir we change for animals purpose.
Nutrient management is more efficient.
In hydroponics, yeah. We are also targeting the nutrient management part so that we can enhance the metabolite production. And we are also integrating the IoT sensor so that we can monitor pH and temperatures. And we don't have to do that manually. We can get the report on our mobile application. We are working on it. Yes.
Good afternoon, sir. Myself P. Bhavna. I am a PhD scholar from OUAT and I have conducted my research work at CRRI Cuttack. So my study basically belongs to like how the volatile or rice plant volatiles can be explored or exploited for plant defense against rice leaf folder, which is a major defoliator. So over-reliance of pesticide in order to manage this pest have led to insecticide resistance, pest resurgence and environmental pollution. So steps have been taken. Now we have thought about eco-friendly and sustainable and management technique which can be included even in integrated pest management technique by using plant-induced defenses in which herbivore-induced plant volatile, which is a part of induced plant defenses, can be explored for pest management techniques. So let us see that preliminary studies have been done, sir. Screening have been done, taking 52 red rice accessions, including susceptible check and resistant check. Susceptible check is TN1 and resistant check is TKM6. So based on the preliminary screening of 50 red rice accessions in 3 field screening and 3 net house screening, like in field screening I have taken 2 Kharif season and 1 Rabi season, and in net house screening 2 Rabi seasons. So based on screening we have found that 2 highly resistant varieties, that is Asopol and Caliban, and 2 highly susceptible varieties, that is Kalobad and Kauka. Along with this 4 varieties, along with the checks, we have taken for volatile extraction. Volatile extractions were done in both the uninfested condition and after infestation with rice leaf folder and allowing it to infest for 4 hours. Then again we have taken for volatile collection. And after taking this volatile, crude volatile from both the
All lab experiments.
lab experiments, sir.
Not yet.
No, sir. So initially we have to identify those compounds, sir, which will act as an attractant or repellent to this rice leaf folder. Based on that, we can take for further semi-field conditions. So in identification, first these crude volatiles were injected into GCMS machine for identification. Yes, sir. Compounds. So based on those identification through GCMS from all the crude extracts from resistant, susceptible in both the healthy and uninfested in rice leaf folder infested condition, we have found 52 compounds. So on the basis of 52, metabolomics was done. In PCA biplot, we have observed that most of the compounds, that is terpenoids, aldehydes, these are the compounds responsible for these are highly induced or highly present in uninfested resistant, infested resistant, and even uninfested resistant, infested susceptible even share some of the terpenoids and aldehydes. Whereas in a very contrasting, we will observe that uninfested susceptible is present in right hand side of the PCA biplot, whereas uninfested resistant is present in the left hand side. So more of the alcohols and esters are enriched in uninfested susceptible. And based on that, we have further dived into PLSTA, that is supervised data, in which in VIP score we observed that the compounds scoring more than 1 are highly responsible for separation into different treatments. Okay. So based on that, methyl jasmonate, R-limonene, xylene, these are some of the aromatic and terpenoid compounds which are highly enriched in infested resistant and uninfested resistant compounds. So we have come to a understanding that terpenoids and aldehydes, these are some of the compounds. These are highly present in resistant compounds, which primarily may be contributing to resistance. So based on that, we have taken study and we have evaluated those crude extracts. So because of space unavailability, I have only pasted uninfested susceptible rice variety crude extracts, in which GCMS and GCEAD was simultaneously run, where we have observed that the positive deflection is of GCMS and negative deflection as inset is neural nervous system is negative deflection. So at every RT or retention time of the compound in GCMS, which is rising above, the insect is giving some response. In some cases, insect is not giving some peak. So we observe that when there is a sharp peak coinciding with the GCMS, so we can understand this is a response. So taking the 15 replications, we have concluded that uninfested susceptible rice genotype, taking the from Calobath, Cauca, TN1, these are the common 31 volatile organic compound which are responsive to splice leaf folder. Similarly, sir, I have done for resistant in both uninfested and infested condition. So I have got common 42 compounds, and 42 compounds I cannot take further for behavioral studies. I need to screen it down. So for screening, I did electron antinogram, in which in a pipette we have placed filter paper and 100 microliter of individual synthetic compound was injected, and the same EAD setup was there, and we need to observe what are the magnitude of EAG amplitude for all those 42 compounds, and we observe that higher responses or higher EAG amplitude was observed for alcohol, then aldehyde, then least was for terpenoids and ketones, these are compound. So based on that, based on EAG magnitude, alcohols such as even esters such as cis-3-hexanyl acetate, 1-octene threol, hexanol, these hexanol, 1-octene threol, these are having higher magnitude of EAG. Based on that, from 42 we have screened down to 21. So these are 21 compounds where we have done behavioral study. In behavioral study, we have taken white tube olfactometer. It is a Y-shaped olfactometer. In one arm it is treatment, another arm is control. In treatment, we will keep synthetic compound which will be injected into filter paper, and moth will be allowed. If the moth travels, like we will take 15 replication with different 15 rice leaf folder moth. If the number of entry of moth is higher in treatment arm, it will be considered as attractant. And if this doesn't move to treatment arm, or it doesn't or move towards control, it will be considered as repellent. So based on that and based on statistical significance, it was observed that trans-2-hexene-1-ol, cis-3-hexanyl acetate, 1-octene threol, 2-hexanol, xylene, these are highly attractant compounds. Whereas beta-carotene, methyl jasmonate, beta-osmin, these are repellent compounds. So sir, this is just a preliminary study. It has not been done like stabilized. Sir, for taking into field, we need to stabilize this compound. So further research needs to be taken down. And sir, we can take synthetic compounds, sir. Sir, even I have done my one objective. I have used different concentration, and in that in greenhouse conditions, and it was observed that beta osmin and beta carotene, like ovipositional preference is very low. Like if it is sprayed on TN1 plant, TN1 is a susceptible variety. When these compounds are sprayed at 200 ppm, these are acting as a repellent almost similar to TKM6. But sir, because of greenhouse, I can change the dispenser every day. Yes. So this needs to be stabilized, and further it can be further taken for HIPV module, in which push-pull techniques are like in undesirable crop we can use the attractant, or we can in susceptible variety we can spray the repellent compounds, and this is how we can reduce the rice leaf folder infestation and destruction. Thank you, sir.
Good afternoon, sir. Myself J. Vinayak, SCTO from Indian Institute of Soil and Water Conservation, Koraput, Research Center, Koraput. This is sir बायोफ्लॉक जो है ना सर, बायोफ्लॉक फिश फार्मिंग क्या होता है ना, जैसे तो हम मछली पालन करते हैं, बायोफ्लॉक में क्या है, हम फिश कल्टीवेशन विथ माइक्रो ऑर्गेनिज्म, बेनिफिशियल माइक्रो ऑर्गेनिज्म। जैसे हम हमारा जो पॉन्ड में अगर फिश कल्चर करेंगे तो 8 महीने वेट करना पड़ता है, इसमें हम 100 डेज में फिश प्रोडक्शन लेते हैं। और उसके लिए सिलेक्टिव कुछ जैसे कोरापुट में हम किए थे, कोरापुट में किए थे सर। कोरापुट क्या है ना, उसका सॉइल रिटेंसिटी, वाटर रिटेंसिटी कैपेसिटी भी मिट्टी का नहीं है और स्लोपी है, पानी नहीं रुकता है। तो ये जो टेक्नोलॉजी ये वहाँ पर जैसे छोटा टैंक में मैंने किया था सर, 20 फीट 10 फीट और 5 फीट में 700 तिलापिया छोड़ा था, गिफ्ट तिलापिया छोड़ा था। वो गिफ्ट तिलापिया से मेरे को 350 किलो मिला था विद इन 100 डेज। उसमें 303 क्रॉप किए हैं हम लोग वहाँ। ये तो हमारा ICAR का है ऑर्गेनाइजेशन। एज ए फिशरीज ग्रेजुएट में दूसरा ऑर्गेनाइजेशन में भी ये ट्राई किए थे, बहुत लोग अडोप्ट कर रहे हैं, तो बहुत बेनिफिशियल है। लोग छत के ऊपर भी इसे रूफटॉप में कर सकते हैं, आपका घर के पीछे भी कर सकते हैं। थोड़ा सा कंटिन्यूस इफोर्ट चाहिए। थैंक यू सर। ठीक है सर।
हाँ हाँ,
कैसे प्रोडक्ट कच्चे को रखते हैं तो पकता है?
नहीं सर, नॉन कोलाबरेटिव। हाँ
फिर भी अगर सिनेसेंस जो होता है
यस सर।
सड़ना या हो सकता है इसकी वजह से वो कम होता है।
हाँ
2-4 दिन 5 दिन हम ज़्यादा इसको रख सकें,
हाँ, वो तो है इफेक्ट सर।
वो इफेक्ट है।
हाँ।
वो फिर प्रैक्टिस में है।
हाँ नहीं सर,
मैंने एक्सेसरीज की बात करूँ, जैसे आपने ब्राउन ऑक्सीडेटिव वही चीज़ हो गई।
हाँ हाँ,
एक तरह से
मेनली सर एंथोसाइनिन का रिटेंशन ये अच्छे से कर पाता है, और भी है।
तो वही है इसके एक्सेसरी फायदे जो हैं, जो आपके अलग अलग कॉन्सेंट्रेशन हो सकते, थोड़ा सा और कॉन्सेंट्रेशन बढ़ा के आप देख सकते हैं कि
नहीं तो तो सर हार्मोनिक रिलेशन आया है, 1 में सही आया, फिर उसके बाद डिटोरिएट हो गया।
डिटोरेशन
हाँ, और 2 में तो ऑलमोस्ट बहुत ज़्यादा डिटोरिएशन आ गया था, मतलब फायदा नहीं हो, कलर डिग्रेड हो गया।
स्किन का ठीक है, स्किन का इतना ज़्यादा आपका फ्लैश अच्छा होना चाहिए,
हाँ
healthy
हाँ
होना चाहिए, रिटेन करना चाहिए।
हाँ।
जो चीज़ें आप करते हैं स्किन काला होने से क्या पता
नहीं
ठीक है,
सर,
मार्केट एक्सेप्टेबिलिटी ठीक है, लेकिन लगता है प्रोसेसिंग इंडस्ट्री के लिए,
हाँ हाँ
के लिए जब आपने जो रसगुल्ले बनाए हैं
हाँ हाँ हाँ, उसके लिए
सब
बेस्ट है।
चीज़ के लिए तो फायदा है। फ्रेश आपको कितना आपका जाता है।
पहले तो सर सल्फाइटेशन वाला था, तो उसमें जो बार्क के द्वारा डेवलप किया गया था, तो उसमें हमारा 180 डेज तक था चल रहा है, रेफ्रिजरेटेड कंडीशन में था, पर पूरा उसका कलर मेंटेन रहता था। लेकिन अभी तो वो सल्फर को बैन कर दिए हैं एक तरह से उसका।
एक वहाँ पे स्टॉल लगा हुआ है इसका रेडिएशन का।
अच्छा सर, लगाए हैं।
रेडिएशन कॉस्टली इक्विपमेंट है,
हाँ
लेकिन
हाँ
इसके लिए हो सकता है रेडिएशन में देखिए उसको एक्सपोजर देखिए।
सर, हम लोग लास्ट टाइम ट्राई किए थे इरेडिएशन करने का, नहीं कर नहीं पाए सर। वो डिफरेंट डोजेज में इरेडिएशन पे BCKB में इरेडिएशन का प्लांट है सर पूरा, तो बार्क का ही है। तो हम लोग वहाँ से कोलैबोरेशन के लिए पेपर कर रहे थे, पर फाइनली वो हो नहीं पाया इस बार।
ये डेस्कटॉप है,
हाँ
मतलब ये कमरे में रख के
हाँ हाँ हाँ
उसमें रेडिएशन कर सकते हैं।
हाँ
इसमें अगर रेडिएशन का फर्क पड़ता है तो उससे आपका जो बोरर
हाँ
ये जो पोस्ट है वो डैमेज है, वो भी कम हो सकता है।
सर बोरर में सबसे बड़ा दिक्कत है वो जब फ्रूट सेटिंग होता है ना वो हाँ
सेटिंग में क्या देता है, बच्चा देता है क्या
अंडा देता है
अंडा मारता है,
हाँ हाँ
अंडा कब निकलता है, हैचिंग कब होती है,
सर जब फ्रूट राइपनिंग के टाइम पे हाँ
पहले
हाँ हाँ
रेडिएशन से या मेलाटोनिन से या कोई भी उसमें इस तरह का जो इम्प्रेगनेट कर सके जो केमिकल,
हाँ हाँ नहीं तो वो तो है सर केमिकल है वो जो हमको नाम नहीं पता सर पर स्प्रे करके उसको हम कंट्रोल कर सकते हैं
अंडे को यूज करने के लिए
हाँ
अंदर होता है, स्प्रे काम करेगा, अंडा अंदर होता है,
हाँ हाँ तो
स्प्रे से नहीं
इरेडिएशन सर कैसे करें एक बार अगर तोड़ लिए तो ख़त्म मतलब वो फिर पकेगा ही नहीं ये लीची
हाँ तो पके हुए
पक हाँ एक बार जब फ्रूट मैच्योर हो गया तभी उसको हम हार्वेस्ट कर सकते हैं तो वो हाँ मैंगो में वो एडवांटेज है ऑरेंज में बहुत सारा एडवांटेज है पर लीची
जितना चाहिए अच्छा काम करने हो जाएगा।
इसमें सर अभी एक हम लोग ट्रायल किए थे वो एक पैकेज सिल्वर इन्फ्यूज्ड एक मॉडिफाइड एटमॉस्फेरिक पैकेज एक कंपनी के साथ यूफ्लेक्स है एक कंपनी से उससे हम लोग दे के मंगवाए थे तो उसमें सर 5 डेज तक लीची बहुत अच्छा था बस वही है कि इनिशियल क्वालिटी थोड़ा देख के रखना पड़ रहा था अगर गड़बड़ फ्रूट चला जा गया जिस पैकेट में वो गड़बड़ हो गया कोई ट्रीटमेंट नहीं कुछ नहीं सर बस उस पैकेट में डालिए एयर टाइट उसको सील कर दीजिए और उसमें माइक्रो परफोरेशन है तो वो ना मॉइस्चर एक्यूमुलेट होने देता है ना ज़्यादा फ्रीज ड्राइंग पे सर लास्ट अभी इस बार हम लोग थोड़ा सा एक ट्रायल किए थे हमारे पास तो ड्रायर है नहीं सर तो मखाना में
फ्रीज ड्राइंग बहुत है किसके लिए,
हाँ हाँ सर
किशमिश बनाएंगे ना
हाँ 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 सर
उसमें वो बनाना पड़ेगा।
हाँ
चलिए ठीक है बात होती रहेगी।
ठीक है थैंक यू सर आपका तो नंबर है
लाइव चल रहा है, लाइव चल रहा है, तो लाइव चल रहा है, लिंक आपको भेज देंगे। खोलो 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 तो
Can I take one? Can I take one? Thank you. Thank you.
কলেজ হবে না কোডা কলেজ অডিওটা নিন না এটাতে তাদের কল লাগে বার করে